Hello, everybody. It's Mr. Cavanis here. In our last lesson, we talked about the regulation of gene expression in prokaryotic cells, E. coli specifically. We were talking about how the cell controls when genes are switched on, when they are transcribed, and how genes can also be switched off and not transcribed. And of course, what we're really talking about here is regulating whether or not a particular protein or polypeptide is being made. Because if you switch a gene off and you don't transcribe it, then you're not ever going to do translation and you're not going to build that protein. But if you switch a gene on and you do transcribe it, you make that messenger RNA, then the ribosome is going to attach to that messenger RNA and build the protein. Now, as you might expect, when we talk about eukaryotic cells, that's going to be our focus today, the regulation of gene expression is considerably more complex. Most things are more complex with eukaryotic cells, but we're still kind of talking about the same thing. We're talking about how cells control whether or not a particular gene is going to be transcribed and translated. In other words, whether or not a particular protein is going to be made. And in some cases, maybe how much of that protein is going to be made. When we regulate gene expression in eukaryotic cells, that regulation can occur at different points in time, different levels, you could say. In other words, we can regulate whether or not a gene is transcribed. So we're going to talk about that. After transcription, there's some regulation that can take place affecting the messenger RNA and how long that messenger RNA is allowed to exist in the cytoplasm, which affects how many times a ribosome has an opportunity to translate it. So you can regulate genes before they're transcribed, and in eukaryotic cells, you can also regulate genes after transcription, before translation. And then even after translation occurs, there are things that can happen to the protein after it's made that affect the functionality of that protein, that affect how long that protein exists inside the cell. So as I said, regulation of gene expression in eukaryotes is pretty complex, but we're going to break it down. We're going to make sense out of it. And today's lesson is going to be a little different. I'm going to introduce it. I'm going to go over some basics with this recorded lecture. I'm going to try to be short, as brief as I can be. But then the details are going to be filled in by a man named Mr. Tate. Robert Tate is an AP biology teacher in Tampa, Florida at HB Plant High School. And I've got a lecture that I'm using with permission from him, and I'm going to let him fill in some of the details after I do a brief introduction. So here we go with the introduction. So all organisms must regulate which genes are expressed at any given time. In a multicellular organism, the regulation of gene expression is an essential part of a process called cell specialization. When our embryonic stem cells begin to specialize and give rise to liver cells and skin cells and nerve cells and blood cells, that process of cell specialization is very much linked with the regulation of gene expression. Because every cell in your body liver cells, muscle cells, brain cells, for example, they all have the same genes, the exact same DNA, the exact same set of genes. But different genes are expressed, that means transcribed and translated, in a liver cell than are transcribed and translated in a muscle cell or a blood cell or what have you. Okay, almost all the cells in an organism contain an identical genome. This is what we were just talking about. Differences between cell types, differences between nerve cells and muscle cells, for example, result from differential gene expression. The expression of different genes by different cells that all have the same genome. Abnormalities in gene expression can lead to imbalances and diseases, including cancer. And one of the things I'm really going to focus on here is that gene expression in eukaryotic cells, like your cells and my cells, animal cells and plant cells, can be regulated at many different stages. So this was the illustration from your textbook that shows all the different points. If you look at these little colored boxes, the color-coded boxes, each box represents a different stage in the process where a gene, this would be the gene up here in the DNA, where a gene can be regulated. So there's a lot of different levels to this. Let's just kind of zoom in here. So part of what you're going to learn about is what factors affect how condensed, how tightly packed the DNA is. This is chromatin. Remember, that's the long, thin, coiled up strands of DNA. That's how DNA exists in the nucleus most of the time. Well, if you've got some genes that are not needed, genes that do not need to be expressed, one of the easiest ways and most basic ways to keep those genes turned off is to keep that whole area of DNA very tightly coiled up. If that chromatin is very tightly packed, then RNA polymerase is not going to be able to get in there and attach to the promoter to do transcription. So if you want to switch off a gene, just keep the DNA tightly coiled up, and it's physically impossible for that gene to be transcribed. So we're going to learn about some of the factors that affect how tightly packed or how condensed the chromatin is. Multiple factors affect how easily RNA polymerase attaches to the promoter. We've already talked a little bit about this back in the transcription lesson several weeks ago, but specifically here you're going to be learning about transcription factors and how they assist in getting RNA polymerase attached to the promoter so that transcription can begin. Now, once a gene is transcribed and that DNA code is rewritten in the form of an RNA message, messenger RNA, there are lots of things that happen to that RNA. We know that the 5' cap is added. We know the poly A tail is added. You're going to learn a little bit more about the importance of that cap and tail. We also know that the introns in a eukaryotic cell have to be spliced out. And when the exons are spliced together, that editing process can take place in different ways. Remember, we talked before about alternative RNA splicing. So you'll be learning more about that. The structural organization of chromatin packs DNA into a compact form. That's what I was talking about right here. If the DNA is tightly packed together, then those genes in that particular area are going to be switched off. But what you're going to learn that's new here is that chemical modifications to the histone proteins. These are the very tiny proteins that DNA wraps around in the nucleus. And chemical modifications to the DNA itself can affect how tightly packed the chromatin is, and therefore affect whether or not RNA polymerase excuse me, can attach to that DNA and start transcribing genes. Now you're going to hear a lot in Mr. Tate's lesson about heterochromatin and euchromatin. So let me just kind of introduce those terms. Heterochromatin is a big fancy word that just means DNA that is highly, highly condensed, tightly packed, which means it's difficult for RNA polymerase to get in there and physically attach to the promoter to express that gene in that area. The opposite of that is euchromatin, which is an area of DNA that's not as tightly packed. It's a little looser. It's less condensed. And so it's physically easier for RNA polymerase to get in there and bind to that DNA to start the process of transcription. So one of the main things that I want you to really listen for when you watch Mr. Tate's lesson is what are the factors, what are the things that happen that cause DNA to be in the form of heterochromatin, and what are some things that happen to cause the DNA to be less tightly packed, which means it's in the form of euchromatin. So I'm just going to introduce that just a little bit. One of the things Mr. Tate is going to talk about is histone acetylation. And I'll let him explain a little bit better what a histone is. But basically a histone is like a spool that DNA can wrap around. I think I've got a picture. Yeah. So these little purple spheres, little purple dots that you see, those are the histone proteins. And you can see the DNA, which is the blue strand you see here, is wrapped around those histones like little spools. Well, the more tightly wrapped the DNA is, the more tightly packed this is. That's heterochromatin. That's DNA that we cannot transcribe. But if we want to be able to transcribe the DNA, we need for that tight knot to loosen up, so that we've got some areas where the DNA is exposed and it's euchromatin, and the DNA, the RNA polymerase can get in there and do transcription. So how do we do that? Well, acetyl groups are little chemicals. They're clusters of atoms. You can see it's a couple of carbon atoms and 3 hydrogen atoms and an oxygen. The whole thing is called an acetyl group, and it has a negative charge, you see right there. So there's a process called acetylation where these little acetyl groups are physically attached to histones, and it makes them become slightly more negative. And DNA is already negative, so if you make the little purple histones negatively charged, and the DNA is already negatively charged, two negatives don't like each other. They repel. And so that will cause the DNA to unwrap and look more like you see it here. These little green dots, by the way, those are the acetyl groups. So if you add acetyl groups, you cause the DNA to unwind and become euchromatin. Now, if that's confusing, it's okay. You're going to get another lesson on this. Mr. Tate will explain it in more detail. Now, the opposite of that is to chemically modify the DNA, not the histone, but the actual DNA, so that it wraps up more tightly, so that it becomes more condensed and harder to transcribe. This is what you would do if you wanted to shut down the expression of a gene. So this is called DNA methylation. It means methyl groups. This is what a methyl group looks like. It's a carbon and 3 hydrogens, so it's 4 atoms. A methyl group can be added to DNA. It's actually added on to the C's in the DNA, the cytosines. And the more methyl groups you add to the DNA, the more hydrophobic the DNA becomes. And anything inside of a cell that is hydrophobic is going to tend to clump. It's going to tend to try to get as far away from water as it can. And so it causes it to sort of clump together in a tight mass, which means the DNA is going to look more like this if it's methylated. It's going to be tightly condensed, and that means it's going to be heterochromatin, which means those genes are switched off. It's going to be hard for RNA polymerase to get in there and do transcription. Individual genes are usually more heavily methylated in cells where those genes are not going to be expressed. In other words, that's one of the main ways we turn genes off in our cells. So to use like a really simplistic example, if I've got a cell in my arm, in this skin cell on my arm, all of the genes that code for eyeballs, or all of the genes that code for the formation of teeth, I want to switch those genes off. Very early in my embryonic development, the genes for teeth and eyeballs, those genes need to be switched off in the cells that are going to become my arm skin. So one of the ways we switch off those genes is to methylate the DNA, make it become hydrophobic, make it coil up tighter, so those genes will be turned off. So once genes are methylated, they usually remain methylated, so they're kind of permanently turned off in those cells. By the way, today's code word for extra credit is Coach Purvis. Coach Purvis. Okay, so an important example, kind of a famous example, I guess you might say, of how methylation can be used to deactivate not just individual genes, but in this case, an entire chromosome, is in mammal females, including human females. As you know, mammal females have 2 copies of the X chromosome. Males, of course, only have 1. So that means every cell in a woman's body, every somatic cell, has 2 X chromosomes. But only one of those X chromosomes is actually needed in each cell. So what happens is, during embryonic development, very early in the life of a female, as the cells are dividing in that little tiny ball of cells, that early embryonic stage, each cell randomly and permanently deactivates one of the X chromosomes. And it's not always the same one. So one cell might deactivate the X chromosome on the left, and another cell right next to it might deactivate the X chromosome on the right. In other words, it's completely random. Now, how do you deactivate an X chromosome? By adding lots and lots and lots of methyl groups. That's methylation, DNA methylation. That causes the entire chromosome to become hydrophobic. So the chromatin coils up really, really tight, so tight that none of those genes on that X chromosome will ever be expressed. A X chromosome in a mammal female that has been permanently deactivated by methylation is called a bar body. I assume it's named after the person who first discovered it. So in each cell of a female's body, only one of the 2 X chromosomes is actually active, and only the genes on that X chromosome can be transcribed. Now, here's a really cool, this is a little off topic maybe, but it does relate to what we're talking about. This is kind of a cool example. In some cat breeds, the gene for fur color, black or orange, is a sex-linked trait. So if it's a male cat, of course he's only going to get one X chromosome. So if that X chromosome has the orange allele, then that is going to be an orange cat, an orange male cat. On the other hand, if a male cat inherits the black allele, which is a recessive allele, on his X chromosome, then he'll be a black male cat. But remember, females get 2 copies of the X chromosome. They're going to always deactivate. Here's the bar body. Here's the deactivated X chromosome. They're always going to deactivate one of their 2 X chromosomes in any given cell. But in female cats, if both X chromosomes have the black allele, then it doesn't matter which X chromosome is deactivated. They both have the black allele, so it's going to be a black female cat. And if the female cat is homozygous orange, again, it doesn't matter which X chromosome is randomly deactivated. They both have the orange allele, so it's going to be an orange female cat. But if you've ever heard the expression calico cats, this is what a calico looks like. It's a cat that has splotches of black and orange fur dispersed across its body. Now the white on the belly and the face and the paws is a separate gene, so that's kind of totally separate issue. But the splotches of black and orange are caused by the fact that this is a sex-linked trait, so only female cats can inherit both the orange and the black allele. And when that happens, some of the cells in that cat embryo will deactivate this X chromosome, the one that's carrying the orange allele, so those cells will only produce black fur. But other cells in the same cat embryo will randomly deactivate the other X chromosome, the one that has the black allele. So that's going to give rise to cells in that same cat that can only produce orange fur. So when those embryonic cells develop into a actual kitty cat, some of the cells in that cat's body are going to produce black fur, and some are going to produce orange fur, and it's going to be random, and it's going to be dispersed over the body of the cat. So other than some really weird exceptions, all calico cats are female. They have to be. And now you know why. Okay. Though chromatin modifications do not alter the DNA sequence, methylation of the DNA, acetylation of the histones does not change the A's and the G's and the C's and the T on the DNA. These chromatin modifications can still be passed to future generations of. So this is kind of interesting. This is a new idea. It's something that scientists are only beginning really to understand. We used to think that the only biological thing a parent can pass down to their offspring is the DNA, the actual A's and G's and T's and C's, the actual code that's in our DNA that we pass down through the sperm cell and the egg cell. Now we know that's not the only thing we can pass down. The inheritance of traits that are transmitted by mechanisms that are not directly involving the nucleotide sequence, this whole branch of study is called epigenetics, or epigenetic inheritance. So we're talking about the idea that you don't just pass down your actual DNA code to your kids, but you're also passing down some of these acetyl groups, these methyl groups that have been attached to your genes. That some of those things, even though they can sometimes be reversed, sometimes they're not reversed and they are passed down to the next generation. And I'll be completely honest, epigenetics was not something I studied in college because nobody knew about it in the 1990s, or at least it wasn't known about enough to really talk about it in college biology classes. It was brand new. And so this is something that I am really learning about for myself, and scientists are really still kind of discovering how epigenetics works. Mr. Tate will talk more about this in his lecture. Some other things that Mr. Tate will touch on is how do we regulate eukaryotic genes after transcription has occurred. So let's assume that we had some euchromatin, RNA polymerase does attach to the gene, transcription has happened, here's our messenger RNA, the RNA splicing, the editing has already taken place, so now this messenger RNA is in the cytoplasm. Okay, so we're ready for a ribosome to attach and start reading the codons and building the polypeptide. How can we regulate this process now? Well, there are factors that Mr. Tate will talk about that affect how long this messenger RNA survives in the cytoplasm. Once it gets into the cytoplasm, there are enzymes, hydrolytic enzymes, that will begin to eat away and destroy this messenger RNA. So it has a limited sort of lifespan. Now, why does that matter? Because if messenger RNA sticks around for a long time, like if it hangs around in the cytoplasm for a long, long time, then that means the ribosome can translate it again and again and again and again and again and again, so we can build lots of this particular protein. But maybe it's a protein that we don't need a lot of. Maybe we just need a little bit of that protein. Then what we would do in that case is we want to make this messenger RNA not last very long. We want it to be destroyed quickly so that it only gets translated a few times. On the other hand, if we need a lot of this protein, then we want to increase the longevity of this RNA. So he's going to talk about some factors that affect that. One of them, just real simple, is if you increase the length of the poly A tail, if you make the poly A tail much, much longer, that's going to increase the length of time that this messenger RNA hangs around in the cytoplasm. You're going to learn how the translation of messenger RNA can be regulated. There are proteins that can attach over here on the 5' end, right beside the 5' cap. There are proteins that can attach right here and physically block the ribosome from attaching. So if you have a situation where you've already done transcription, but you temporarily want to shut down the production of that protein, you can attach these proteins right here and it'll block the ribosome. Let's assume that we're not blocking translation, so the protein has been built. Well, now there are things that can happen. We can regulate this polypeptide. There are things that can happen to it that will affect how that protein folds and what shape it takes on and what kind of job it can do in the cell. And the final level at which gene regulation can occur in eukaryotic cells is controlling or regulating the degradation of a protein. So if we have an active protein that is no longer needed, an easy way to shut down that protein, I mean, you could shut down the transcription and the translation, but let's assume it's already here, it's already been made, it's already active, and now we want to shut down this protein. Well, one way to do that is just to chop it up into little pieces. If you want the protein to stop working, you degrade it. And there's a mechanism in the cell that does that it breaks down proteins that are no longer needed, or maybe they're worn out, and you're going to learn how that process works right at the end of Mr. Tate's lesson. So here's a link to the lecture by Mr. Tate that I was talking about. I'm actually going to post this lesson in the form of an ed puzzle. So you'll watch it, and every few minutes a question will pop up, and we can do like a classwork grade or something just based on how many of those questions you get correct. But probably there'll be a quiz on this, not only on my lecture today, but on Mr. Tate's lecture as well. One of our little 10, probably it'll be one of the little 10 question quizzes that you've gotten used to. So that's it. That's my introduction. Guys, I hope you are doing well. I miss you, and I hope to see you soon.